Is it better now? Yeah, it is. Perfect. So I'm going to present this talk, which is called Development of a Label-Free Approach for Melanoma Diagnosis using Spectral Imaging and Phasor Analysis. This is a project we developed here at the Advanced Unit. It started as my bachelor's thesis. So in this talk, I'm going to introduce a little bit about phasor analysis and spectral imaging, the idea of a model-free project, a model-free analysis, and the protocol we use to apply this model-free, then the application we use of this technique, and finally, I'm going to talk a little bit about the software we are developing that just started with this project. So spectral imaging, as you've been working these days, it's a technique or a pipeline, as we say in computer science, that you acquire a spectral. I'm going to use the mouse to show you here, so the ones in Zoom can see it. You acquire a stack of image, which is a spectral image, and you, for each pixel, you will have a spectrum, like here, a one-dimensional signal. You will apply the phasor transform, which is a kind of Fourier transform, and you split this transform into a real and imaginary part, so you build like a cluster with its dot. In the cluster, you can see the distribution of each spectrum. So somehow you can categorize or see like components that have similar frequency response. Okay. Then we use this is the average image of the stack that we use it to see the shape of the image and interest structure for us. And finally, with the components in the phasor, we can build like a pseudo-color image that relates different components with a color scale. In this case, it's like a spectrum scale that range from red to violets. And then we have some properties that are very useful in a model-free approach, that are the change in the phase and the change in the modulation. If your spectrum has a larger wavelength, the phase is going to be wider or bigger, which is the angle here. Then if the modulation change, also the distance between the origin to the dot, it will change. If the amplitude of the spectrum is bigger, the modulation is smaller. So this gives you an idea how the spectrums are just looking at the phasor and not looking at the spectrum, okay? And finally, the linear combination. Can you hear me on the Zoom? Yeah, perfect. So if you have two pure components, like here, the blue and the green, and you have like the sum of both, this sum is going to be in a linear that goes through green to the blue component. So where did we use this model? We have a problem with the melanoma diagnosis. The melanoma and the nevus are melanocytic proliferations. The melanoma is the malignant tumor, and the nevus is like the benign one. It's commonly known as the mole you have on your skin. And there are like a pipeline also in the diagnosis. The first one is the presumptive clinical diagnosis that it's done by the dermatology at the clinic. They use this. They use the dermatoscopy here to look some characteristic on the shape of the mole, which are if it is asymmetric, if the borders are irregular, if there are changes in the color, the diameter is bigger than 0.5 millimeters, and if it is bald through time. So if they found some of these characteristics, they are going to send you to the surgery, and they are going to remove the mole, and then the anatomopathologist is going to search for some characteristic in the hematocylin eosin image. These characteristics are like thickness, if there are presence of ulceration, cell division, and many others. So they look for it and they make the diagnosis. The problem here is that we don't have like a biomolecular marker, like you can make a test and see if expressed in the presence of something. So the diagnosis ends up being very dermatologist dependent, okay? There are some techniques that are being assessed these days, like multiphoton microscopy, to see if in the melanoma there is a change in the bound FID concentrations. But this is a very expensive technique, as you have seen these days, and it depends on the technician that uses the microscope. It has to be, it's difficult to use it on the clinic too, because you have to use it on the surgery or immediately after they remove the tissue from the patient. So what we propose to use fixed samples, okay, and use spectral imaging with a confocal microscope. So the protocol we established was to use label-free image with our confocal microscope, the 8080, with an excitation at 405. Then we collect the rest of the spectrum, and we will perform phasor analysis. So when we did that, we found some challenge that arise from the sample. The size of the sample are too big. It's required a tiling. You have to make a tile of the entire image and then rebuild it. Then the thickness of the sample, you will focus in a place, and when you are doing the tiling, it will be out of focus in other places. Some microscope has like automatic autofocus, but that gives you another kind of signal. So all of these things were challenges that we managed later, okay? And finally, the acquisition time is very big. Like this sample here that I'm showing you, it's 80 per 14 image, and it took me like 5 hours to do it, and it's only one sample. It is this one here. This is the hematocylin eosin, but I use the label-free. And it's a small one, actually, because if you can compare with the other one, here it's much bigger. And actually, I couldn't do this one. So to the image tiling and the stitching, what we did was a little guided by how the microscope acquired the image. We created an algorithm that takes care of the direction of the scanning and also the percentage of the overlapping. Okay. What was the problem? First, if we don't do that, we will have misclassifying components on the phasor, because when there are like, these are the image S and G, which correspond to the coordinate of the phasor. So when there are high changes in the frequency, like here you have like gray and dark. So if you average that, you will have like this effect in image B, which is like a ghost effect in an image. So we try to optimize this and obtain a better image in the overlapping region. And then the other thing we have to evaluate was the filtering techniques, because you can acquire the same image many times, as we saw on the practical session, and then reduce the signal-to-noise ratio. But the problem here is that it's much more time consuming. So we evaluate some filters in order to improve the quality of the image. So we compare a twice time acquisition image with a 6 time, which is the higher time that the microscope acquire. And then this one we filter with a median filter, and we reduce the scattering of the phasor. Okay, and also we reduce the damage of the sample because you irradiated less, and the exposition time. So what we obtained with that? Well, for the nevus, we use the hematocylin eosin to the dermatologist. She was like analyzing each part in order to compare with our result, and we obtained the label-free spectral image. We compute the phasor, as you can see here. And finally, we create a pseudocolor scale, which range from red to violet. And this is like our final image. When we did the same pipeline to the melanoma, here is the tumor section, which was segmented by the pathologist. The same pipeline, and you can see that there are many other components on the phasor. It has a larger wider phase. So the pseudocolor image shows you where the tumor is. We did that with 10 samples, 5 intradermal nevus and 5 melanoma, invasive melanomas. And we were looking for something more quantitative. So as I mentioned, the dermatologist segmented the area. So we went deep here and look what is going on with the phasor in these areas, okay? Here is like where the melanocytic proliferation is in the nevus, and here is in the melanoma. We compute the phasor for that, and then what we calculate was the modulation histogram and the phase histogram to see the distribution of these parameters on the phasor. After that, we still needed a value, okay? So what we proposed to use was the center of mass of these histograms. We did it with the 10 samples, with the 10 patients, and we acquired the center of mass for the modulation and the phase. And finally, we would like to, since each patient has a modulation and a phase, we created like a cluster and plot both families. And here is where we saw that melanomas and nevus are very separated on this space. Then, as I mentioned, this is a model-free approach. We don't know what it is spectrally, what components are there, but we found a difference. So we already know what is on the skin, okay? There are collagens, NADH, elastin, melanin, FID. So we acquired the spectrums, the pure spectrum of them, to see how they distribute on the phasor. And then we compare it with some regions. We make these regions of melanocytic nest. They are like accumulated, the cells accumulate forming nest. And we compute the phasor. We compare how these pure components are distributed over the phasor. And we saw that all the collagens, the elastin, are more related to the nevus, and the distribution of the components we saw here are related to the melanin and FID. There is also another component pushing here, as you can see, that we haven't been able to characterize yet. And finally, what we saw in this study is that applying hyperspectral imaging and phasor analysis in autofluorescence with a UV excitation have a value in the diagnostic. The main autofluorescers were NADH, FID, melanin, and elastin, also collagen. And finally, the modulation and phase histogram enables us to create a further approach of a quantitative value to differentiate them. Our further perspective with this project is to build a score, a quantitative score, in order to segmentate or differentiate both of them, combine phasor data with AI, also compare the information with 2-photon microscopy and film. This project is being developed by Sofia, the dermatologist, and Lionel. Well, and finally, we are working on the development of a hyperspectral phasor scanner in order to do that speedier and improve the acquisition time. This paper was recently published on Frontiers in Oncology, that you can find it here, this link. And as I mentioned, we've been coding and developing some algorithms to process all this data. This first library, which started in my bachelor's thesis, ended up being granted by CCI, and we get 2 projects. One is the phasor body library, which is a library that will make phasor analysis for film and spectral imaging. In this project, we are working with Enrico, Christoph, and Bruno Panuccio, which is right over there. You can also see the web page in the QR. And the other project was the Napari plugin that we saw yesterday. This is a GUI interface that you can perform phasor analysis only in spectral imaging. It's also in his first stage, but will have his first release soon. This is a project we are working with Lionel. And I would like to thank Sofia, which is the dermatologist, Lionel, and well, the rest of the equipe that's been working with us. Thank you.